Hello students, good evening. Welcome to another session from brilliant Doha, Qatar. In this class, in this video session, today we are going to discuss a complicated chapter, Principles of Inheritance and Variation. In our previous two classes, we have discussed molecular base of inheritance and also sexual reproduction in flowering plant chapter. This chapter, very complicated chapter. In this chapter we will discuss pedigree analysis questions, monohybrid, dihybrid questions, clear, and sex linked inheritance question, mutation question, gene mutations, chromosomal mutations. A lot of new topics we are going to understand. And moreover students, I am requesting you all keep NCERT with you. I am going to give from reference from NCERT. Everyone keep NCERT and open and keep it with you. Okay, and in this chapter a complicated questions are going to come. I will make it simplify, and today I will finish at least 30 questions from this chapter. These questions will help you to brush up your concept before final examination. So all of you, I hope you all are ready. I'm audible to you all. Right, now I'm going to start the first question. Look at the first question. This is pedigree analysis question. Look at the question. Study the pedigree chart given below. Okay, what does it show? It's given. My dear students, keep in mind we can expect one pedigree analysis question in final examination. So pedigree analysis questions come. So there are only 4 different type of pedigree analysis are there, right? One is called autosomal dominant, autosomal recessive. Third one is called sex-linked recessive, sex-linked dominant. Only these 4 are there, right? One is autosomal dominant one, autosomal recessive one, sex-linked recessive one, sex-linked dominant one. There are 4 disorders are there. There are 4 major problems can come. Look at here. In this pedigree, parents are unaffected. There is no affected in the parents, right? F generation. So this is, I'm writing here, it is F generation, and this is F1 generation, and this is F2 generation. Parents are unaffected. No effect in the parents, they are good only. Now come to the next generation. Here son affected, this is son affected, and here it is daughter, right? Daughter affected. Third generation also affected. First you have to carefully think, out of the 3 generation, all generations are affected, or any skipping generation is there? Look at here, F generation did not affected, right? That means what? It's skipping the generation. Whenever skip the generation, we can understand it is a recessive disorder. In case of dominant disorder, at least one individual in every generation will affect. That is the biggest hint, keep in mind. So in this pedigree, F generation, parent generation did not affected. So this is a recessive one. But the second point is this autosomal recessive or sex-linked recessive? That's a problem. We have to find out that also. Look at here. So assume here this is sex-linked recessive. Sex-linked recessive means here this is a carrier. So here it is normal person, that means XY. Here it is carrier, we can say for example XC, XM, what they call, assuming. Here it is. Now if it is a sex-linked dominant, this daughter would not show the disease, right? So it is clear it is not sex-linked disorder. Come to the option. So what is left now? We have a one option. What is the one option? Autosomal recessive condition. Autosomal recessive condition example is what? We can write phenylketonuria, thalassemia, sickle cell anemia. These all are, right? Now look at here. Now inheritance of a condition like phenylketonuria as an autosomal recessive condition, right? It's appropriate, correct? Let me take rest of 3, it's correct or wrong. Now look at here, the next third one. So second one, the pedigree chart is showing wrong, so leave it this option. It's not correct. Now inheritance of a recessive sex-linked disease like hemophilia, this is not sex-linked. Remember, this is what autosomal recessive. So what's the right answer? The right answer of this question is option A. It is a recessive. Which type of recessive? Autosomal recessive. Parents unaffected, children's affected, or offsprings affected. Remember student, when parents unaffected and offsprings are affected, that is the recessive disorder characteristic feature. That is recessive disorder character. And moreover, parents unaffected, son and daughter both are affected in the F1 generation, it will not be a sex-linked recessive. It is not be a sex-linked one. Got my point? So the right answer of this question we can write inheritance conditions like phenylketonuria is autosomal recessive condition. So first question answer is autosomal recessive condition. Next question, question number 2. Question number 2. This type of questions will come. Look at the question carefully. This type of questions will come. Linkage, T.H. Morgan, first time. Experimental proof of what? Linkage. On Drosophila melanogaster, what they call fruit fly. Look at the question carefully. Refer to the given figure. Cross A, cross B. Select the correct statement regarding them. Fine. Now look at here. This is cross A. In cross A, Y gene, W gene they given. Yellow, white body and Y plus, W plus. Here it is cross B, that is W, M genes are given. Fine. Look at the first one. What is the first option is given? First option, in cross A, the strength of linkage between the genes YW is higher than the cross B. WM. So if you wanted to answer this question, just look at the cross A, cross B, the distance between the genes. Distance between the genes. In case of cross A, Y gene and also W genes are closer than the cross B, right? Am I clear, student? Look at the B. B, in case of cross B, W and M genes are little apart. Little, what they call, distance is there between these two genes. Answer me. When distance more, linkage less. Yes or no? When distance more, linkage less. So when distance more, there is a chance of segregation, separation. When distance more, there is a high chance of what? Recombination in the next generation. Am I clear? So look at here. What they're asking, in cross A and cross B, I can tell in the cross A, distance between the two gene is very, that means less. That means linkage is more. Distance is less, linkage is more. So parent characteristic will be repeated in the next generation, right? So majority of the parent characteristics will go in the next generation, right? Why? There will be no recombination. There will be no crossing over. There will be no, tell me, segregation of gene. Am I clear? That's why in NCERT it is given in cross A, approximately 98% of parent generation is repeating. Am I clear? Now look at here. In cross A, what is given? In cross A, the strength of linkage between the genes YW is higher than the cross B, W and M. Yes, it's correct. Keep aside. Keep aside. Let me take the B. In cross A, the strength of the linkage between the genes YW is lesser. YW, look at the YW is not lesser, it is very high. The linkage is very high. It's not correct answer. Come to the C. Both cross A genes YW and also cross B same strength. There is no same strength. Look at here. In cross B, Y as a W and also M genes are apart. So distance is there. So there is no same strength. Am I clear? Come to the D option last. The percentage of recombination produced in cross A is higher. No. Recombination will not become higher. Why? Why recombination is not high in cross A? Come on students. Any one of you? Adil? Allah? Come on, any one of you? Reshma? Why in the cross A recombination is very less or not high? Sir, because the two genes are linkage genes. This was given by whom? This was given by T.H. Morgan. Linkage questions come, remember students, when the genes are close, when linkage is more, recombination less. When genes are apart, when genes are far, when genes are long distance is there, that means more recombination. There is a chance of more recombination. This type of questions will come. Linkage, gene mapping questions come. So everyone go through NCERT carefully. This clear cross A, cross B, F generation, F2 generation, clearly it's given. If you able to recall, in cross B, what is given in cross B? Open your NCERT and look at there. Recombination is 33-34% is given. That means more recombination in cross B because distance between the gene is high. Remember. This type of question. So I can write the answer is option A. First two questions I hope clear. First two question I hope clear. Third question I'm giving to the online students only. You have to answer now. Look at the third question. Down syndrome is caused by an extra copy of chromosome number 21. Fine. It is a Down syndrome. We also call, what shall I call it, a trisomy? Yes, it is trisomy. What type of trisomy? 21st autosomal trisomy. Done. 21st auto. So what is the karyotype of this person or this individual? I can write the karyotype instead of 44 autosome, 45 plus XX if it is female, or 45 plus XY. Am I clear? It could maybe both male and female. Now look at the question. What percentage of offsprings produced by an affected mother, that means female, they are talking about female. Let me write here female, that means 45 plus XX is the karyotype of this mother. Here it is given what mother, this is mother. Fine, mother. What is the karyotype of this mother? 45 plus XX. And normal father. Normal father means they're crossing. This woman is crossing with the normal male. Normal male. Let me hear. Normal male is 44 plus XY. Can you tell me what percentage? What they're asking here? Normal and would be affected by this disorder. How much? What is the percentage would be affected in the next generation? Carefully look at here how I am solving this question. Students, mother 45 plus XX, right? Listen. She will give two different type of eggs. One type of egg is 23 plus X chromosomal egg. This is called egg or female gamete. Next one is 22 plus X chromosome. This is the second egg. One gamete is normal gamete, 22 autosome plus 1 X chromosome, 1 sex chromosome. But one egg is abnormal egg. That means one extra chromosome is a 23. So look at here. Here the male partner, the father is going to give a same or normal sperms. What are the normal sperms? Now 22 autosome plus X chromosomal sperm, 22 autosome plus Y chromosome sperm. 22 autosome plus Y chromosome. I'm going to read the comments. Anyone? Option is given. Yes, approximately. Yes, exactly. It is a 50% chance is there. How 50% chance? Look at here. There are two type of eggs are there, right? If this abnormal egg is crossed with the normal this sperm, tell me now, how can I write now? 45 for 23 plus 22, 45 plus XX female, Down syndrome female. Done. This normal one is going to fertile with the normal one, 44 plus XY normal male child. So what percentage? 50% of chance is there to get what? Down syndrome in the next generation. You should know first the karyotype of this Down syndrome and how many type of gametes she is going to produce. So otherwise you cannot answer this question. You don't know. If you don't know the karyotype, the Down syndrome karyotype, you cannot answer this question. That's why students, everyone, chromosomal disorders are very important. Klinefelter, Down syndrome, Turner syndrome. So I can write what option is 50% of children can be affected. You understand my point? 50% children can be affected. Next question. Question number, I think, 4th one. This type of questions will come. Hemophilia, colorblind, crisscross pattern, sex-linked inheritance question come. This type of questions will come. So look at the question, what the question is telling us. Now the question, a normal woman whose father was colorblind. Normal woman whose father was colorblind. Can you please tell me the genotype of this woman? Can you please tell me the genotype of this woman? I am reading the comments. Raina, Reshma, Dharani, Adin. What is the genotype of this woman? Is she a carrier or normal woman? Is she a carrier or normal woman? No. No, Allah. Father is colorblind, right? So here the father was colorblind. So father to a daughter, this will be spread. Mother to son will be transfer. Yes or no? So I can write what? Tell me here. A normal woman whose father was colorblind, he is married to a normal man. The sons would be, they're asking, what is the possibility of a disease in the son or disorder in the sons? Let me take here. Here it is XC, X. This is, who is this? Who is this? The woman genotype. This woman genotype, right? Cross with normal men, XY. Tell me here, two type of gametes, XC, X. Here it is two type of sperms, X sperm and Y chromosome sperm. Tell me now, first one, XCX, XCX, carrier girl, daughter. Next, what about this one? What about this one? XCY. Who is this? Affected son. Affected son. Next, what about this one? XX, normal girl. Right? Normal daughter, normal son. So here, how many total sons are there? 2 out of 4 individual, out of 4 offspring, 2 sons are there. What percentage of affected? Yes, exactly 50%. So we can write what? 50% of sons are affected. Look at here, 50%. You have to be think carefully, students. This woman is carrier or normal? Am I clear? They given a hint. The father of this woman was colorblind, so she must be a carrier. So when carrier woman marry a normal person, 50% of chance of what they call male child or sons can be affected. Am I clear? So question number 4 is clear, option B. Question number 5. Sickle cell anemia induced to. It's very, very easy question. Sickle cell anemia induced to. I need your answer. I need your answer. Reshma, next Raina, Adin, Dharani. I need answer. Sickle cell anemia. It is an autosomal recessive disorder. Mutation takes place in the beta polypeptide chain of hemoglobin. Which position is that? 6th position in place of glutamic acid. What comes? Valine. Yes or no? Well, it is a point mutation. Yes. Now, change of amino acid in the alpha chain. No, sir. Change of amino acid in the beta chain of hemoglobin. So the answer is option B. So what type of disorder is this? Is this autosomal recessive? Autosomal recessive. Till here everything clear? I am clear. Next question. Next question. Can you do this question? Can you do this question? This type of questions will repeat. So carefully read the question. What the question is given, the hint. What is the hint here? One of the parents of a cross has a mutation. Mark this one. Mark this one. Mutation in its mitochondria. Mitochondrial genes. Whatever the mitochondrial genes are there, mitochondria DNA mutant. Okay, which mitochondria? In that cross, parent is taken as a male parent. Male parent mitochondria mutant. I have a question. One small question from the human reproductive system. Answer me. Which part of sperm DNA is there? Which part of sperm DNA is there? 2 places DNA is there. One is called head part, haploidal 23 chromosome DNA is there. Second place also DNA is there. Where? Mitochondria. Where is the mitochondria present in the sperm? Neck region, right? Head part, neck part, tail part. Am I clear? Now look at here. Let me take the sperm. Okay. Here it is. This is secondary oocyte. This is secondary oocyte. Haploidal nucleus is this one, right? Now this is sperm. Look at it. This is sperm. Right, this is sperm. Head part is here. This is called acrosome. Done. We have learned in the fertilization topic which part of sperm only entered into the egg. Which part of sperm? Only head part, right? Only haploidal nucleus of sperm only deliver into the egg, right? Now look at here. This haploidal nucleus, 23 chromosome, haploidal nucleus only entered into the cytoplasm. Now this is haploidal nucleus of egg, right? Haploidal nucleus of egg, haploidal nucleus of sperm fused to form zygote. Zygote. This is called fertilization. Answer me. Mitochondria are present in the middle piece. The mitochondria are not going into the egg. They are not participating in where? Fertilization. They are not participate in the fertilization. So if a mutation in the DNA of mitochondria also, no problem. Because we will not inherit, we will not get the mutant DNA from a sperm, from male partner. That means father mutant DNA of mitochondria, if it is there also, no problem, because father mitochondrial mutant DNA is not participating in fertilization. Sir, here this is egg. In the egg also mitochondria are there. This mitochondria having what, tell me? This is mitochondria, and this mitochondria having the DNA. But this DNA belongs to mother, right? Because it's coming from the female partner. Am I clear? You get my... This is called cytoplasmic inheritance, students. Cytoplasmic inheritance we will only get from the maternal, that means mother, not from father. But in the question, the mutation takes place in the male partner. That means mutation in the mitochondria of sperm. Mitochondria of sperm is not participating in the fertilization. So what's the problem? So pick up your answer. What is your answer? What is your answer now? So 1/3 of the progeny will affect, none of the progeny affect, all the progeny affect, 50% of the progeny affect. So what is the answer? What will be the answer if the fertilization, if the DNA, mitochondrial DNA is not participating in the fertilization? None of them. So we can write answer is option B. This type of complicated, tricky questions can come. You must be very careful. Are they asking about the DNA of head part of sperm or DNA of mitochondria? DNA of mitochondria is not participating. You must know this. Mitochondria sperms only used to give energy for sperm to move towards. When the time comes, a fertilization time comes, it will not participate in the fertilization. So none of the progeny. Next question, question number 7. Sex determination, one of the complicated and very important concept from principles of inheritance. Sex determination, one question can come. Sex determination, male heterogamety, female heterogamety, XX, XY, ZZ, ZW, ZZ, Z0. This type of questions can come, students. So look at here carefully. ZZ, ZW type of sex determination seen in 1 minute. Everyone look at here. Female, heterogamety, female heterogamety, two types. A, B. One is called, tell me, so one is called, here I'm writing now XX, not XX, it is ZZ, Z0, ZZ, ZW, ZZ, ZW exclusively seen in where? Buds. Fouls. Look at the question. Look at the question. ZZZW. That means female partner is going to determine the sex of offspring, right? What is option 1? Option 1, platypus is a mammal, snail, cockroaches and peacock bird. Option D. Option D. Yes, good, very good. Till here everything clear. And in case of insects, XX, X0. In case of human being and Drosophila, XX, XY. This ZZ, Z0, few type of moths. Am I clear? I'm going to the next question. Question number 8. In an individual affected by phenylketonuria, autosomal recessive inborn metabolic error. Inborn error of metabolism. As it's a problem of gene. One particular gene is going to be mutant, point mutation. So there is no synthesis of a particular enzyme. Phenylalanine hydroxylase, hepatic enzyme secreted by liver, essential for the converting one amino acid to other. What is the amino acid name? Look at here. Phenylalanine to tyrosine. Remember all of you. And an individual affected by the phenylketonuria lacks an enzyme. What is the enzyme here? Phenylalanine hydroxylase enzyme. That enzyme converts amino acid dash into dash. They are asking. Phenylalanine hydroxylase enzyme convert a particular essential amino acid phenylalanine into tyrosine. So what is the right answer? The right answer is option B. This tyrosine is essential for brain growth, brain development and the other processes. Thyroxine hormone releasing, this all are. In case of phenylketonuria individual, phenylalanine hydroxylase enzyme absent. So if phenylalanine hydroxylase enzyme absent, phenylalanine amino acid does not convert into tyrosine. It convert into phenyl pyruvic acid, phenylalanine pyruvic acid. That pyruvic acid accumulated in the brain to cause mental retardation, low IQ. These all are the characteristic feature of what? Phenylketonuria. Phenylketonuria is the example of pleiotropy. We discussed this one, pleiotropy. One mutant gene can control many traits. Clear? Am I clear? So the right answer is option B. Question number 9. Any one of you do this question, very easy. Match the following. What is the first option? What is the first option? Now look at here. Dominance. What shall I go? Many genes govern a single character. In a heterozygous organism, only one allele express it. In an heterozygous organism, both allele express. A single gene inherited influenced by the many character. Dominance. Dominance genes mean, we can write here for example capital A, dominance. Capital A, small a also dominant. So out of two genes, one gene will be a dominant gene. So I can write, good. I can write dominant mean in an heterozygous or only one allele will be expressed to. What is codominance? What is codominance? Any one of you online student, what is codominance? Codominance mean here, I can write the codominance is here. Heterozygous organism, both allele will be expressed. Both allele will be expressed. Example is what? AB. Blood group IA, IB, both will be expressed. Next, pleiotropy. Many genes are governed by single characteristic feature is a polygenic. Polygenic, many genes, many genetic, polygenetic inheritance, human skin color and human height. Many genes involved in one trait. So I can write last one is pleiotropy. A single gene influence many characters. Just now only we discussed phenylketonuria example. So final answer is 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, 1. Option is D is the right answer. Am I clear? Next question. Question number 10. This type of questions can come. Remember all of you. Look at here. It's very, very important question. Look at here. Number of Barr bodies in XXY male. Can you please tell me XXY disorder is called? XXY disorder is called Turner, Down, Klinefelter. Which one of these three? Klinefelter syndrome, right? This is Klinefelter syndrome or not? Look at here. It is a trisomy. What type of trisomy? Sex chromosome trisomy, or I can also say allosomal trisomy. Done? Remember student, everyone look at here. Barr body means extra X non-functional chromosome. There are, look at here, let me take what I'm explaining. Here it is, in case of males, XY chromosomes are there, right? One X chromosome is there, that functional throughout the life. So there is no Barr body. That means the normal 44 plus XY. This condition, that means normal male, I can write without Barr body. Remember, zero Barr body. Come to the normal female, X44 plus XX. 2 X chromosomes are there. 2 X chromosomes will be functional till embryonic stage. Am I clear? Early embryonic stage. After that, one of these 2 X chromosome will going to be inactivate. That inactivated extra X chromosome, this X chromosome is called Barr body. Barr body. So how many Barr bodies in normal female? 1 Barr body. One is functional. Out of 2 X chromosome, one is functional, one is non-functional. So extra, not that, whatever the non-functional chromosome is there, that we call the Barr body. So normal females have 1 Barr body. Normal females have 1 Barr body. I am writing Turner syndrome karyotype. Okay, I am writing what? Turner syndrome. 44 plus X0. This is a Turner syndrome, student. Turner syndrome. So this Turner syndrome, female without Barr body. Normally, normal females have 1 Barr body, right? But this is, who is this? This is female. Which female? Turner syndrome female. But there is no Barr body. In case of normal male, look at the normal male without Barr body. Look at here, this is Klinefelter syndrome. 2X chromosome is there. One of this X chromosome, non-functional X chromosome. I can write this is Barr body. Remember all of you. It is male. Normally, males do not have a Barr body. But in this condition, Barr body is there. So how many Barr body? Choose your answer. How many? Yes, absolutely. Barr body, one Barr body. So I can write the answer is one Barr body. You will get this type of question. I will write something on the board. Can you please tell me how many Barr bodies? For example, I am writing X, 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 female. Can you please tell me how many Barr bodies? In this genotype, in this karyotype, how many Barr bodies? Can you? Reshma, Adin, anyone? Abin, Abdul? Yes, 2. Exactly 2. Look at here. One is a functional. These 2 are non-functional. So I can write the 2 Barr bodies. 2 Barr bodies. Am I clear? Am I clear? So based on this one, you can select that how many Barr bodies given the karyotype. Clear? Next question. Question number 11. This, look at here. Incorrect statement. Incorrect statement they are asking. Select the incorrect statement. Human males have one of their sex chromosome much shorter than the other. Much shorter than the others. Come here. We know 22 chromosomes, up to 22 chromosome, autosomes. Fine. Autosome, 22 pairs. 23rd pair of chromosome, sex chromosome. This 23rd pair of chromosome, one chromosome is bigger, longer, second chromosome is shorter. Whatever the shorter chromosome is called Y chromosome, and longer chromosome is called X chromosome. This statement is true. So this statement is correct. Male fruit fly heterogametic. Yes, fruit fly the given Drosophila XXXY. Right? Next, in male grasshopper, 50% sperms have no sex chromosome. Statement correct, because X0 type of sex determination. X0 is males. X0 is males. Correct? Look at the option D. I told you this one earlier. In domestic fowls, birds, sex of progeny depends on the type of sperm. No, type of egg. Because the birds are showing what type of sex determination? Female heterogamety. So here they are talking about what? The male partner, the sperm will decide the sex of the progeny or sex of the offspring. No. The female bird, the mother bird will decide. So incorrect statement is option D. How many of you given correct answer of this? Very good, Sana. Good, very good. Next one. Next question. Question number 12. Which of the following is an example of pleiotropy? Can you guess this one? Which of the following is the example of pleiotropy? Answer. Any one of you? Example of pleiotropy. Absolutely correct. Very good, Dharani. What's the right answer here? Phenylketonuria. Phenylketonuria. Out of the 4 options, 2 are sex linked. Can you guess what are they? Color blindness and also hemophilia. These 2 are what? Sex linked. Next question. Question number 13. You have to do this question very careful. Look at here what they given. My dear student, if you open the NCERT textbook, the backside of questions, there was one small question is given. After I explain this question, I will come to that one. Look at here. Whenever the genotype is given like this, whatever the genotype is given, what is the genotype they given? Let me write here. The genotype is capital A, capital B, small b, capital C, small c. Can any one of you tell me how many heterozygous are given here? How many heterozygous are in this genotype? This is what? Genotype. Can you please tell me how many heterozygous are there? I need answer. How many heterozygous? 2 heterozygous are there. Adin, not 4 beta. It's 2. 2 heterozygous, not 4. Look at here. 2. This is heterozygous condition. This is also heterozygous. There is a formula. 2 power n. What is n here? N equal to number of heterozygous. Now here in place of n, 2. 2, 2. Look at here. What is the number of gametes can come? 4. How many different types of genetically different gametes will be produced by the given heterozygous? This genotype is given. This genotype, if you know how many heterozygous are there, answer is very easy. So answer is what? Option 4. Am I clear? What NCERT is given? The NCERT the backside of question, heterozygous for 4 loci. 84 heterozygous is given. So 2 power 4, you can calculate the number of number of gametes. Understand my point? So you have to think how many heterozygous given in the genotype. Based on that one, you can pick up the right answer. Question number 14. I need answer of this one. All of you take pen and paper. Come on, all of you take pen and paper and do this cross. A cross between capital A small a, capital A small a, capital B small b, cross with small a, capital B. Any one of you tell me from this genotype how many different type of gametes can come? I am not asking how many number. I am asking how many types. How many types here? Any one of you? Because here one heterozygous is there, so 2 different type of gametes can come, right? 2 different type of gametes can come, right? Because one heterozygous is there. So what are the gametes now? Tell me here. I can write small capital A, capital B. Am I clear? Small a, capital B, small a, capital B. Now what about here? All 4 are or all are small a, capital B, small a, capital B. So find out your answer. What is the right answer? What is the right answer? Now if you cross between these 2, there is a possibility of anyone. Now look at it. This one. Now I capital A, small a, capital B, and this one. What is that? Small a, capital B. Choose your answer. Choose your answer. The right answer of this question. Right, very good. The option A. 1:1 ratio. This type of dihybrid cross questions come, student, carefully read here. You should know how to take the gametes, otherwise the complete ratio will wrong. Next question. Question number 15. Can you do this? I will wait for 1 minute. Can you do this? Any one of you online student? Both husband and wife have normal vision, though their fathers were color blind. Given hint. We got a hint. I don't know what is that. You have to answer me now. Can you do this? Then Shah, next Raina. Can you do this? Reshma, Dharani, Angeline. Can you do this? Both husband and wife normal vision. That means they don't have a color blindness problem. Okay, I can write here X cross with X Y, but their father color blind. That means XCY. Father, here look at here, they given what here? Father cannot transfer this disease to son, so son is normal here. Father can transfer the disease to or that affected gene to a girl, that means daughter. So here I can write this woman is carrier. Am I clear? Now can you do it now? Now 2 gametes can come from here, XC, X, and from here also X and Y. They are asking what? Colorblind. The probability of the daughters become colorblind. Daughter become colorblind. XC, X here. No colorblind here. Right? Next here it is. What about the son? XC, Y. There is a chance is there. Now here XX, no daughter affected. XY, no son affected. That means our answer is what? Our answer is 0%, right? Am I clear, online student? Answer me. 0%, right? The daughters are not colorblind. Their carrier daughters are there, but there is no affected individual. So our answer is 0%. Am I clear? Are you with me? Is it clear till here? Everything clear? Right. Next question. Question number 15. Inheritance of skin color. Simple direct question. Skin color, human height, human skin color. What type of inheritance? Simple question. Very good, absolutely. Polygenic inheritance. What is polygenic inheritance? One trait, that many genes are controlling one trait, single trait. Human skin color, capital A, capital A, capital B, capital B, capital C, capital C. There are 6 different genes. I told you, when dominant genes number increase, pigmentation also increase. Am I clear? Now, next question. This type of questions look at here, carefully look at here what they're asking. Sickle cell anemia, glutamic acid is replaced by valine. Fine. Which of the following triplet codon for valine? They're asking. Let me take here the problematic gene. Okay. Now, problematic gene is what actually? GUG. Right? What is the template code? Template stand C A, and here it is C. What is the coding here? G T G. Done. This is problematic one. What is the normal one? Let me write here G A G, and what is the template one here? C T C. And what is the normal mRNA codon? Normal mRNA codon, I can write what? G A G. What happened? Carefully look at here. 6th place, glutamic acid. This is a normal HbA person hemoglobin gene. This A mutant to T, my dear student, this is important. A, adenine is mutant to thymine T. Because of this problem here, this is coding strand, okay, and this is because of here it is T, automatically the template stand also will change. When template stand change, now look at here, mRNA in place of here it is, what comes in place of A? U comes GUG. GUG code for valine, GAG code for what? Glutamic acid. Open all of you, insert and cross check. So the right answer is 6th place of beta polypeptide chain in hemoglobin. Instead of glutamic acid, 6th position valine comes. So answer is GUG. Am I clear? Till here everything clear? This question, this type of questions will come, student. You have to be careful. What is the normal? What is the mutant one? Tell me here. HBS carrier, HBA normal, HBS sickle cell anemia. 3 genotype, possible 3 genotype. One will be sickle cell anemia. Next question number 18. Segregation of Mendelian factor capital A small a occurs. Carefully, 1 minute, look at here and listen. This type of questions, Mendelian alleles or Mendelian factors or alleles they can give, or you can also get homologous chromosomes. My dear students, if you have a little knowledge in the cell cycle, cell division, meiosis 1, look at here what I am writing, look at here all of it. This is chromosome, homologous chromosome. On homologous chromosome, genes are the alleles, capital A allele, and here it is small a is there. Done. Now, after metaphase, in the anaphase 1 of meiosis 1, remember all of you, anaphase 1 of meiosis 1, what happened? This homologous chromosomal pair, homologous chromosomal pair, if I am writing like this, look at here, this is cell, here it is centriole, from centriole what comes? Spindle fibers, like this. Then we have learned this one in the 11th grade. These are called asterase. Here also asterase are there. This homologous, out of 2 homologous, everyone remember, these 2 homologous chromosomes are started to separate. Segregation of homologous chromosome in anaphase 1 of meiosis 1. sister chromatids remain associated but homologous chromosome apart. So what happened now? Look at here carefully. This chromosome, this is homologous chromosome here, right? And whatever the next homologous chromosome is there, this homologous chromosome started to segregate here. That means out of these two chromosomes, one chromosome go to one side, one pole, other chromosome come to another side. Homologous chromosome separation in anaphase 1 of meiosis 1. Now, this Mendelian factors, these alleles are located on homologous chromosome. So when homologous chromosomes are segregated, automatically the gene also will go, right? Look at here, when this chromosome is going here, this complete chromosome is going, A also will go, A gene also will go. This complete chromosome is going, small a also will go. So alleles also separating. So write down here, diploid, no, anaphase 1. Remember this type of questions. Anaphase 1 of meiosis 1, they separate. Am I clear? Next question. Can you do this? A fruit fly is heterozygous for sex-linked gene. Fruit fly. What type of sex determination? XX, XY. I'm writing here XX and XY. Sex determination. Fine. Now, when mated with the normal female fruit fly. Normal female fruit fly. Let me write here like this. Okay. Now, this is XY cross with normal. This is a normal fruit fly. This is mutant one. Okay. The male specific chromosome will be entered into the egg cell in the proportion of, I am giving chance to you. Anybody? Anybody? Out of the 4 options, right? Very good. Right? Any one of you? What proportion of? Exactly. What about others? What about others? Allah, Dharani given, Shah and Reshma. Yes. Now tell me here. Now it is a problematic gene. Let me represent this problematic gene is here. I can write it is what? Tell me 1:1, right? 1:1 ratio will be. So answer is option C. How simple question it is. 50-50 percentage. 50-50 percentage. Next question. Question number 20. This question, everyone pay attention. 2 minutes, everyone pay attention. Gene mappage questions. Mapping, gene mapping question. What the question is asking? There are 3 genes A, B, C. Let me take a chromosome here. Chromosome, okay. On the chromosome, there are 3 genes are given. A, B, C. Percentage crossing over. A, B is 20%. Let me take here A, and what I'm writing here it is B, 20%. Between these two it is 20%. Okay, just assume like this. It's correct or not, we will discuss later. And BC 28%. BC 28%. Let me write here it is C. Between here to here 28% is there. Next, and AC is 8%. AC is 8% is given better. Now I can write this one. If it is chromosome, let me write here. This is chromosome. This is chromosome. First what tell me? First what tell me here it is AB 20%. AB 20%. So I can write here it is, this is B and this is A if I write. Between these two, AB 20%, 20% I am writing here. Fine. BC 28%. That means C must be here. C must be here. BC 28%. Next, AC 8%. That means between these two 8%. So the right option is I can write what? A, B, A, C. Can you please tell me which out of these 3 genes more recombination can form? More recombination can form between these 3 genes. Anybody? Any one of you? More recombination. So wherever the distance is more recombination. Yes or no? Wherever the, very good. Wherever the more recombination, where is the more recombination? Recombination between the BC is more, so there is a recombination. The distance between the BC is more, so recombination also more. So can you please tell me out of these 3 genes, which genes are tightly present? That means less recombination. which all the genes are very closely associated? Any one of you, online students, any one of you, which are closely associated, which are close? AC, very good, very good. AC, that means the linked genes. Percentage, 8%. The percentage, the recombination also very less. This type of gene mapping questions can come, student, carefully you have to prepare. Next question, 21st question. A human male produce sperms. You have to answer me now. You have to think and answer me. A human male produce sperm with the genetic genotype of 1, capital A, capital B. Next, capital A, small b. Next, small a, capital B. Next, small a, small b. Four different type of what will make genotype. Genotype of gametes. Genotype is forming. What is the genotype of that sperm? Any one of you? Any one of you? So what is the corresponding genotype of the person? I told you when 4 different type is coming, mean there are two heterozygous. There must be a 2 heterozygous. I hope you got me. I hope you got me. Very good. So I can write capital A, small a, capital B, small b. What about the first one? So here it is 1 heterozygous. So 2 different type of gamete. Here it is 2 different type of gamete. Here it is 0. That means only 1 type of gametes are coming. That is capital B, capital A, capital B, capital A, capital B, capital A, capital A, capital B. What about here? Capital A, capital B, capital A, small b. Next, capital B, small b. Yes or no? Small a, small. 4 different type of gametes. So answer is option D. Absolutely good. Very good, student. You are following me. Next question. Question number 22. I will be silent. I will be silent. You have to answer this one. I already explained in this one earlier. Come on, answer me. Come on, come on, come on, answer me. Sania, come on. Next, Sana, Allah, answer me. Select the correct statement from the ones given below respected to dihybrid cross. Dihybrid cross, correct statement. Tightly linked genes on the same chromosome show higher recombination. Is this statement correct or not? Is this statement correct or not? No, sir, it's not correct. Adin, very correct, very good, very good. The answer is correct. But statement 1 is tightly linked genes, same chromosome show high recombination. No, no, no. Tightly linked genes are less recombination, less recombination. Fine. Genes far apart on the same chromosome show very few very high because they are very far. So easily crossing over takes place in the packaging. Easily crossing over takes place, easily segregation takes place. Genes loosely linked on the same chromosome show similar recombination. How it is similar? Next, tightly linked genes on the same chromosome show very few because they are not going to segregate. So parent characteristics are coming, repeating into the next generation. Am I clear? No recombination. So I can write or no recombination or few recombination. Option D, absolutely correct. Good. Very good. Next question. Simple question. Simple question. I don't want to waste the time. Anybody? Any one of you give me the experimental proof of. Very good. Who is this? Very good. Flip flop. Good. Very flip flop movement in where? You know, the chapter called cell is the unit of life. What is that? Fluid mosaic theory. Good, very good. Now, experimental proof is what? T.H. Morgan, absolutely. On Drosophila. T.H. Morgan on Drosophila. Next question. When dominant and recessive allele both express, what is this? Simple, basic question. You must give the answer. You must give the answer. And Hadia, can you give me Adin? Right, simple question. When both alleles are expressed are codominant. Simple, no need to waste time. Codominant, option A. Am I clear? Next question. Can you do this question? I will be silent. This is the simplest question and a very easy question. We are learning this from 10th grade. We are learning monohybrid cross from 10th grade. Can you do this answer? I will be silent. I will wait here. Come on. Hadia, Dharani, Sania, Adin, flip-flop moment. Come on. What about this? A tall true breeding garden pea. Let me take here. Tall, capital T, cross with small t, F generation. We know F1 generation is capital T, small t, F1 generation. Cross with capital T, small t, cross with capital T, small t, capital T is to capital T, small t, capital T, small t, small t, small t. So what is the right answer? What is the right answer? Yes, exactly. A simple answer. What is that? 3:1 tall dwarf. They're asking genotype ratio, right? Self with genotype ratio. They did not ask a phenotype ratio. Option A is a phenotype ratio. So what is the right answer? Answer is 1:2, 1:2, 2:1. Tall homozygous, tall heterozygous and dwarf plants. Answer is option C. Next question. 26th question. This is a blood group question. Can any one of you do this? Any one of you do this? Look at the question. Says a genotype of husband and wife, IA, IB, okay, cross with who is this and A. Wife is IAI. A blood group. I will be silent. Come on, pick up the right answer. Take pen and paper, Shah. And what is this? Adin. Come on, Reshma. Take this one. Now I can write here two type of gametes. IA, IB. Fine. From here I can take IA and small I. Right. Now what about this one? I can write IA. That means A blood group. Fine. Next, what about this one? This one is IAI, again A blood group. Now what about this one? IA, IB, which blood group? AB blood group. And what about this one? IBI, B blood group. Can you please tell me how many phenotypes? How many phenotypes? How many phenotypes? How many phenotypes? How many number? I need number, phenotype number. Phenotype, very good, absolutely, flip flop, very good, three phenotype, A, B, A, B, A, B, A, B, A, B. How many genotypes? How many genotypes? How many genotypes? Answer me, how many genotypes? 4, right? 4, right? 4, yes, look at here, 1, 2, 3, 4. So pick up the right answer. Pick up the right answer from this option. Now here I am writing 4 genotype, 3 phenotypes are option B. Am I clear? Option B correct? Correct, right? Look at here, 4 genotype. This all are for genotype and 1 is phenotype. Absolutely. Let me take the next question. Next question. Oh my God, this type of graphical diagrammatic, this type of questions, come carefully. Look at here. Disease or disorder transfer from one where to where? I will be silence. Come on, you have to. Reshma, come on. Adin, look at the question carefully. In the question that tells us female, mother to son, it's going, father to daughter. Sir, this is sex linked inheritance, sex linked recessive. Simple. When father to daughter, mother to son, sex linked recessive. Pick up the sex linked one here. Sex linked one, I can pick up the sex linked one. Phenylketonuria. No sir, this is autosomal. Hemophilia. Okay, fine. Sex linked one. Sickle cell anemia also autosomal. Thalassemia also autosomal. So what left? Hemophilia. Am I correct? Done. Very good. Absolutely. Next question. Simple question again, what they call sex linked inheritance question. Can you do this? Take a pen and paper and do this question. Both parents are carrier for thalassemia. I'm sorry, this is not sex linked, this is autosomal thalassemia. Both parents are thalassemia carriers. So I can simply write capital A small a cross with capital A small a. Sir, why did you take this capital A small a? Because it is autosomal recessive. Autosomal recessive you can take like this, no problem. Actually the HBA, what particular gene is there, but you can write it in simple capital S because the carrier, right? The carrier means heterozygous, the parent must have heterozygous. So when you do cross like this, it is thalassemia is autosomal recessive. I have a question, student. Do you remember, until and unless two recessive genes present, disease will not express? In case of autosomal recessive, tell me, if two recessive genes present, only disease will express. Tell me now here. So you will get capital A, capital A, capital A, small a, capital A, small a, small a, small a. So what percentage? 25, sir. Answer is option A. Answer option A, absolutely correct. Last question I'm going to show. Now, can you pick up the right answer? Last question. I am reading. I don't know. You have to say it is. If it is right, right, sir. Correct, sir. If it is wrong, wrong. Now, pick up correct statement. Hemophilia is a sex-linked recessive disorder. Correct or not? Yes, it's correct. Down syndrome is due to aneuploidy. Right or wrong? Aneuploidy. Correct. Very good. Done. Phenylketonuria is an autosomal recessive gene disorder. Correct. Correct or not? Yes, true. Very good. Sickle cell anemia, X-linked recessive disorder. Correct. Correct. Am I correct? Correct. This one also correct. Sickle cell anemia, X-linked recessive also correct. Answer me. Sir, sickle cell anemia, how come it is sex-linked? Sickle cell anemia, autosomal. Sex-linked anemia is what? Autosomal, right? So what is the right answer now? What is the right answer? Option I can tell 1, 2, 3 are correct. So option B. Option B. Right? Shall I show one more question? Will you wait? One more question shall I show? Time up, but one more question. If you are staying here, I will show one more question. Shall I? Last question, one more, last question. Shall I? Right, one more question. Now look at it. The last question. Last question. A woman with 47 chromosomes due to 3 copies of chromosome of 21st. How simple How simple? Can you answer me? Very good. Absolutely. Adin, flip-flop. Very good. Very nice. It's called what? Tell me. 21 chromosome is Down syndrome. Turner syndrome, monosomy. Monosomy. Right? And this is called a trisomy of 21st chromosome. Am I clear? Right. Right. These are the questions from Principles of Inheritance and Variation. I hope these questions help you to get a good marks in your final examination. Thanks for attending this session. Next class, we are coming with Human Reproduction System. Human Reproduction chapter. Important question I'm going to. And a session reason next class I'm going to focusing on. Thanks for attending brilliant YouTube live classes. Thank you. Good night to all.
Brilliant Qatar, your trusted coaching partner for IIT, JEE, NEET, Science and Commerce tuitions with 10 years of excellence in quality training. Brilliant Qatar.